So in my first talk, what I'm going to do is I'm going to talk about using the wisdom of crowds to investigate a disease that I didn't have any patients to, I didn't know anything about before somebody posed the problem to me, but it's papillary renal cell carcinoma. And as I do this, what I'm going to try to do, like I said, is I'm going to basically, as I demonstrate how we use the crowds to investigate a disease, what I'm also going to do is demonstrate some of the software that we also use to do this. So I'm going to be jumping back and forth between the slideshow and then some of the applications. So this all started, has anybody heard of these hackathons that they do on disease? Yeah, that's kind of really popular now. So they get a bunch of computer people, some engineers, some doctors, and they all put them in a room and say, figure out this disease. So, and then they've made some pretty good advancements doing that. Somebody brought this to my attention. Actually, the one that put this one together, Bill Paceman, contacted me through my YouTube channel and said, hey, I've seen some of your kidney cancer stuff. Can you do the same thing with papillary renal cell carcinoma? I was like, yeah, probably. So I work with Illumina quite a bit. I talked to some of the Illumina people and they said, yeah, why don't we do this? Let's see what we can discover using just some of our software, getting a bunch of data sets on papillary renal cell carcinoma and see what we can find. And again, like I said, this is getting popular. I just saw this a little while ago. You know, they're using it to detect, schizophrenia genes now. Again, the crowd is huge. So before you get into any project, you got to figure out, what the heck am I looking at? So, I did some research on kidney cancer, right? And actually the majority of it, so there's many different types of kidney cancer, but the majority of the cancer is clear cell. And the different types of cancers depend on where that cell type is that develops the cancer. But the papillary type's about 15% of all kidney cancers. You have this type 1, I wish my pointer worked on here. You have type 1 kidney cancer, about 5%. It's usually associated with mutations in MET, the MET gene. They look very different than type 2 under the microscope. And not only that, but it's actually a milder form of the disease. It's very curable. Type 2, on the other hand, looks very different cell type, different genes that are mutated. In this one, it's fumarate hydratase that's usually mutated in papillary type 2. And the prognosis is horrible for the patients. Here is why we need bioinformatics for this, is because the oncologists treat both these cancers the same, right? And they're obviously different, right? So what can I use? Can I use all the information that's out there to kind of define papillary renal cell carcinoma, maybe divide these two cell types? And not only that, but maybe give me targets that I can go after. Actually, not go after, my collaborators can go after. And, again, people have done, research on papillary renal cell carcinoma. This was a study done, some pretty large studies. This was in New England Journal of Medicine. And you can see that. Is there a laser pointer here?
There should be one.
Oh, yeah, cool. So we can see this was a study done in New England Journal of Medicine, but again, you have two types, and they're basically defining all these types by the types of molecular, either mutations or messenger RNA clusters, or actually, in this case, methylated units. So again, they find that the three types, but what they find also is that they can subdivide type 2 into three different types of its own, A, B, and C. And if we look here, and no one would care whether it was different types or not if it didn't have some relevance on physiology, right? If we look here, the type 1, as you can see, the survival is pretty good. Again, this is a 100% survival, 0% survival, and this is x-axis time. So again, type 1, pretty survivable. This type 2A seems to be very much like type 1 from a molecular standpoint, or at least from the survivability. Then you have type 2B, which seems to be kind of in the middle, and then type 2C, which is horrible. If you get this disease, prognosis is not good. So that's what my goal was, is how do I take all this information and basically, so what I wanted to do was basically find everything I could on papillary renal cell carcinoma. And to do this, you can actually go into Geo and download the individual studies. Has anybody tried that? Yeah, what did you think of that?
It was kind of confusing.
Yeah, it's horrible. You can say it. It's absolutely horrible. Like, our government puts together the worst data analytics possible. I think it's just to confuse everybody so they get the grants. I don't know. So what I want to do is, so I have this software. This is a correlation engine. This is from Illumina. Everybody has heard of Illumina, right? No? Oh my gosh. So Illumina makes all these sequencers, and they sequence probably 95% of everything on this planet. If you know somebody at Illumina, make friends with them, because they'll probably end up owning the earth at some point. So they have all this data, and somebody at Illumina had the bright idea. It's actually James Flynn, who I know, decided, well, let's put all this information together and use it as kind of a database that all these scientists can use. Not only can they go and get data on their own, but they can use that to compare their data to everything that's out there. So at this moment right now, they have about 23,000 different studies in correlation engine. There's about 100,000 studies in Geo. Why do you think that's different? What do you think? So again, Geo, where we're putting all our data, there's about 100,000. This correlation engine, which basically collects all the information out there, has about 23,000. Why do you think it's less? Yeah.
Is there data that's excluded?
Yes, correct. There's a lot of garbage out there. And in fact, 75% of all the stuff in Geo, they can't even use for this database, right? That's what also makes this nice, is that it filters the data for you so that you don't have to go in there and see if the data is good or not. Okay, so this is what I'm going to do, and I'm going to actually show you some things on correlation engine. So this is a base-based correlation engine. Again, this is a repository for lots and lots of omic data. And there's microarrays, there's RNA-seq, there's all kinds of stuff. So here's what you do is you can just put in your query term as I want to investigate kidney cancer. And as I type, certain phenotypes will come up. Again, what makes this special is not only have they basically collected all the data, but it's annotated really, really well so you can search it, right? So I can hit kidney cancer. And this will just give you some, even just on a phenotype, it'll bring up genes based on its data set that are involved in kidney cancer. Here's that VHL. This is actually mutated in clear cell. But I want it to be more specific. So I want to look for, so I'm going to go to curated studies. So this is where all their data is. If I look here, there are 146 studies that have kidney cancer as one of their keywords. That's a lot of data, right? Obviously, I don't want to look all of that right now, so I can actually filter that even further. So I can filter it by organisms. I can say I only want to look at humans. There's 139 of those. I want to look at RNA expression. And I'm going to tell you right now, if you really want to understand a system, you have to look at the gene expression, right? That I've seen so many cancers with the same, what do they call it, driver mutations? God, I hate that term. Please don't use driver mutations. Same driver mutations, but the phenotypes are very, very different, right? It's what the genes are expressing is that makes the cancer what it is, right? It's not the mutation status, it's not the genetics itself, it's actually what the genes are doing. So we can do RNA expression, and then I can put in a keyword here. And I'll say, I want papillary. And there's a lot of papillary stuff, so I'm not going to select anything. I'm just going to hit apply filter. Okay. Now we're at 11 studies. And this is very easy. This is very manageable now, right? What we can do is we can basically create a meta-analysis. And if you look at this window here, this green looks like a funnel, This is their meta-analysis tab. And right now I have 17 studies in there. So I can look down here. So here are all my studies. You can read about it. You can get full study details. You can add it to your project. But if I select this study, you'll see a negative here, right? So this list, papillary renal cell carcinoma, poor survival versus excellent survival, there was about 6,000 genes that changed. I now own that list. That's now part of my meta-analysis. And we can just keep going. And as you can see, I'm just picking everything with papillary renal cell carcinoma. And if I add something here, so it was 17, now it's an 18, right? This is how easy this is. And that's kind of what you want, you know? As a biologist, I don't want to spend a lot of time programming. I don't want to like learn code. I just want to get into the biology. And this is what allows you to do that. Yeah.
So when you're compiling like a list of studies in this database, how are you normalizing the data within each of these studies?
I'm going to show you that.
Okay.
Yes. And that is key, right? So when you look at microarray studies, so this is basically based on hybridization, and these are these gene chips I was talking about. Your fold chains, actually, microarrays are way better, more accurate than RNA-seq. Most people don't think, understand that. So, you know, the microarrays, the fold changes are pretty lower compared to like RNA-seq, which when you're actually measuring the transcript itself. So you can get huge, you know, you can have 1,000-fold, you know, fold changes. How do you normalize that? And this actually program will do that. So basically I'm using this to collect my metadata. I just went through here. There's 17 studies that I'm interested in. I go to my meta-analysis page. And here are all my studies that I used, right? So all of these are papillary renal cell carcinoma versus either normal kidney or normal adjacent kidney. And there's all different kinds of types, except the last three, it's types of the same cancer compared to itself, okay? We can look at the gene results on that. And this is how, I'm sorry, and this is how I figured out how this stuff works, right? You know, here are all these genes. The odds of having, say, TNF AIP6 differentially expressed in all 17 of our studies is astronomically small, right? This stuff works. And not only that, is when we actually look at, you know, the studies where they compared to normal tissue, it's all going up. So in this instance, if it's a red bar, that means it's increased in that study significantly. And they use a p-value of 0.05 to define that. And it has to be at least above or below 1.2 or negative 1.2 fold change. But you can see this amazing consistency that, you know, one group, and they're all measuring it at different kind of, I mean, it's all high, but they're all measuring it at kind of different levels. And we can actually click on that and look at these. So here are all our studies. Here's the p-values associated with that. And then here are all the fold changes. You'll notice, though, that this fold change here in this one is 9.6, and in this one, which was actually done with the RNA-seq study, is 50-fold. Yet when we actually look at the boxes, this box is higher than this box. So what they do in order to normalize all this data so we can all compare it together is they take the highest fold change and they make that 100. And everything is a descending scale to 0 from that. So everything's at the same scale. Does that make sense? Got it. Right? So now I have all this gorgeous, gorgeous data. And this is more data. This is millions and millions of dollars worth of data that I did not have to generate myself. Right? Everybody sees the advantage of that? Good. All right. So now what we can do is we can export this information, right? So what this program will do is it will take all these genes and all this fold change, the p-values, and the normalized score that Illumina gives it from 100 to 0, and we'll put it on an Excel spreadsheet. And I will show you that right now. me see. We'll do this. No, there we go. So when you download it, this is kind of what you get. This is the Excel spreadsheet you get. Again, you have all your biosets here. Then if you look down, you can see here's all your genes, information on those genes, the amount of studies it was differentially expressed in. There's an overall gene score. But again, you can see here's the p-value in that one study, here is the normalized score, and here's the full change. What I can do now is I can just take all these values here, this normalized score that Illumina gives it, and basically either make it positive or negative based on the full change. So that is my data set. Okay, so we have this data. Now what we can do is we can use other programs to actually look at it. Here's our studies that we collected. Again, here's the criteria for all the genes. So here you can see, in this one, these are the number of test subjects they used. Here's the number of control individuals, and here are all the genes that were differentially expressed. And again, all these genes have a p-value of less than 0.05 and have an absolute fold change greater than 1.2. Okay. Again, I showed you that page where we look at gene expression. You know, that's what we can do is, you know, you can just do it by just look at it gene by gene, right? We can look down here. You know, what is something that's actually, you know, lower in all the studies, or actually has the same pattern in all the different studies? If we look here, the first one that pops into my mind is this dopa decarboxylase, DDC, right? So I looked that up. I'm like, well, you know, we've got this gene. It seems to be dysregulated or lower expression in all these different papillary renal cell comparisons. Does it have any physiological effect? And sure enough, when I go into another program, which is called Cohort Analyzer, that has in their database 881 kidney cancer patients, and if we divide them up by either high DDD expression or low DDC expression, we see there's a huge change in survivability. That if you have high DDC, you're more likely to survive than low. We now have another target, right? Something we can look at. Again, what I'm kind of showing you here is that, you know, this isn't just numbers, and that when you pick a target, you better make sure it has a physiological response. And sure enough, this one does. And so these are kind of the things that we do to kind of, you know, as we go through this data set, to just kind of reassure ourselves that we're on the right track. Okay. And again, now that we have numbers, again, we have that normalized fold changes. I use another program called Partek Genomics Suite. Has anybody heard of that? Yeah, cool, wow. Did you have a question?
Yeah, I was wondering, is it Illumina who hires people to look through the studies and then tag them appropriately?
Yes.
Or is it like a computer filtered?
I think they use a computer a lot, and then there's a human obviously associated with that to make sure everything went correct. Yeah. Yeah, they have a whole crew that's basically all their job is to just take data that they find and put it into their database. So it's always increasing, which is nice. And it's maintained, which is really nice. So again, we can do this. We can do like a heat map. So if that gene had a low expression in that particular comparison, here's all our comparisons up here. Each lane or column represents a different study. If it's blue, that means that gene went down in that particular study. If it's the more intense the yellow, the more that gene went up. And once we put all this stuff together, and remember these last three columns are cancer compared to each other. Again, look at the consistency. But we can see, if we look at the columns that have the type 2, again, we're going to ignore type 2A because based on that study, it's pretty much like the type 1. But if we look here, and we look at this kind of column here, we can see that they're very different. And that actually, when we compare type 2 to type 1, we can see these genes are much more elevated. Not only to type 1 itself, but to basically the control tissue. And so we can pick out clusters in here. My argument is that if a gene, and we're going to show this, if a gene behaves the same way, if a bunch of genes go up or are decreased or increased, over many different studies in the same pattern, they're probably related, right? This particular cluster has, these genes are much higher in type 2, which is the more aggressive type. We can pull these out. We can zero in on them and basically get all of these genes. So here's what all of these genes are doing in all these different papillary renal cell carcinoma studies. So I can take these genes. What we can do now is we can throw it through another program. Has anybody heard of String? Search tool for the retrieval of interacting genes and proteins, I believe. I might be wrong. How do you like String? Yeah, if you're looking at it, only, I think the maximum is, was it like 250, 300, something like that? Something like that, yeah. But if you have under 250 genes, it's a great program. So not only do you need to crunch the numbers, but visualizing it, I would much rather look at this, something like this, than a table. And what we're showing here is each line represents some piece of evidence that connects that particular gene to that other gene. The more lines, the more connection, more evidence of that connection. We can go through here and we can actually color it based on biological functions. And we find that there's, especially with lots of different types of cancer, lots of DNA repair involved, and we're seeing things that we would expect to see. Mitotic cell division, all this cell cycle stuff. This is basically the cancer turbocharged to divide. And then we can pick up different things. We see this whole pool of folate. This is actually to make nucleotides. This thing seems to be upregulated. Maybe that could be a potential target for this disease. And one of these in here actually, and then what we can do is we could say, okay, given all these genes and all these connections, so the odds of having this connection is less than 1 times 10 to the negative 16th. This isn't random chance. What I'm showing you now is basically probably the real true signaling network that type 2 actually goes through. That these genes all interact together to perform that, to make that tumor more aggressive. The gene that had the most connections is topoisomerase 2A. Has anybody heard of that? Okay, well, if you're in cancer, you probably have. But again, so then we can investigate that. Does that have any clinical effects? And sure enough, if you have low topoisomerase 2A, you seem to do much better or survive much longer than somebody with the high type, right? Not only, there's another graphing program. This one is actually commercial, so String was free. So definitely use String. This one is Pathway Studio. It's Elsevier. So anytime you're asking for a manuscript and you're behind a paywall, it says, oh, you have to register. It's going to be $50 for you to see this paper. It's these jerks. But the thing is they have an amazing database, right? They have all this information. So what you can do is you can put this and say, based on your database, take all these genes that I just showed you, the type 2, and graph them out. And what you find is that most of these are in the nucleus, right? DNA repair, cell cycle, it makes sense. But again, we're getting some things on the outside that might allow us to target this type 2. And this one in particular, I know you can't see that. Hyaluronan-mediated motility receptor. It's a long name. And I didn't know anything about this, but when you actually start looking it up, it makes a lot of sense with cancer. And people are showing this associated with many forms of aggressive cancer. Maybe we target this, right? Not only have I put all these genes together, but I found something on the outside of the cell that interacts with all these genes, which might be an excellent target for this cancer. And we can do, so I haven't showed you the cluster of this, but I mean there's a group of genes that are higher in type 1 that seem to define that, and they produce a very different cluster, right? Now you're not seeing so many things in the nucleus. What you're seeing is all this stuff on the outside, this immunological stuff. And again, you see this MET. That is a gene that's mutated in the majority of these type 1 cancers, and you can see it's right in the middle of all that. Or we can take our type 2 signature and we could put it back into Correlation Engine to see what's going on. I'm trying to be on time. So again, we can take our list those genes that were upregulated in type 2, and then we can compare it to all the different studies. And what's interesting is all these breast cancer studies come up. Here's one that's very much like, it's ductal carcinoma versus lobular. That makes sense, ductal. Kidneys, are very ductal. So if something works in this particular cancer, maybe it might work in ours. And we get more information by comparing it. That ours is more like the estrogen negative than the positive forms of breast cancer. If you actually do, you look in papillary renal cell carcinoma, males are, I think it's 5 times more likely to get this cancer than females. And maybe this is why, right? This is some of that evidence. We can find the genes that are common to those and actually investigate those as well. And what this is also showing is that our profile is more like stage 4 than stage 1. So again, it's giving much more evidence that this panel that we just identified is associated with aggressive forms of not only kidney cancer, but other forms of cancer. Does everybody understand that? And here's the real thing with medicine, is that, so not only does this data set have access to all these different studies, but all these studies use different compounds in their studies. So they have a pharmacoatlas. So what you can do is you can put your list of genes in and say, can you find me chemicals that either positively or negatively regulate the genes of interest? And I found one in here. If you actually look in this list, the papatinib comes up. And they actually use that on other forms of cancer. I think it's lung cancer, actually. Do you know? Anyway, this seems to affect a lot of the genes that we saw associated with type 2. Maybe we use this in, say, a cell type and see if it works, right? Take a type 1, type 2, compare. Or we can just do normally, right? We can take the average of all these different studies and do kind of a wisdom of crowd, right? And to generate the idealized form of papillary renal cell carcinoma. So again, that's what I did. Just take the average fold change, normalized fold change of all these different studies, and now we have a master list. Okay, so we can take that list and put it back into our study. So I actually have it saved here. I can save all these different gene lists. So I go to my data, go to wisdom of crowds. So I have cancer versus control. So it was my top 2,000 genes that were common to all those. And now here's my study. And I can go and I could look at the pharmacoatlas. I could say what, regardless of type 1 or type 2, are there any chemicals that would have a negative impact on my system? And we can filter it by that. We can rank these. Maybe. Come on. There we go. Filter it, just look at negative correlation. And so now I'm basically generating a list of things that I think might be a potential therapy for papillary renal cell carcinoma. And we can try all these things. Actually, this second one is awesome. This is Imodin. It's been used, it's traditional Chinese herb. It's been used for thousands of years. Maybe this herb might be, a potential therapy for papillary renal cell carcinoma. And what's also nice too is they have all this access to these knockdown studies. So we've pretty much knocked down every gene in mice, right? And those studies are out there. So if you knock a gene in mice, it has an effect on certain genes. So what they do is they can look at those studies and say, when I knock folliculin out, I think that's folliculin. Yep. It seems to have a positive effect on my genes of interest, which would seem to tell me that this folliculin is probably a tumor suppressor. Does that make sense? Right? I knock it out, all my genes go up. And we can look at that. We can see that study, get more information, check out how they're doing that. So here's my study. I have about 2,000 genes. They have about 8,000 that were differentially expressed. Between the common between those two lists is about 1,000 genes. The odds of that happening are 1.8 times 10 to the negative 56th. It's the same odds of Trump resigning. I told myself I wasn't going to get political. Okay. But again, you can see that when you change, genes in their study that tend to go up in ours. Genes that go down in theirs tend to go down in ours. These are the only genes that don't follow that pattern. The majority of that pattern shows the same as ours, so therefore it's a positive effect. Does everybody understand that? Cool. All right. Okay. So, that's what we're doing, right? So that's basically, how do we use Wisdom of Crowd to look at a basically disease? And it's putting all this information together and finding patterns. You don't need a PhD to see that those genes in that line act very differently across all those samples, right? And they seem to be higher in the more severe forms. Therefore, I bet that is a very important cluster that we need to look at. All right, so now what happens if you're actually looking at just the gene itself?